You're sitting there right now, probably assuming that your body is something stable, something that simply continues from one moment to the next without much effort. But if you shift your perspective just slightly and look at yourself through the lens of physics, that assumption begins to fall apart, because what you actually are is not a stable object, but a dynamic system constantly resisting collapse, continuously pushing back against one of the most fundamental laws of the universe, the second law of thermodynamics, which states that all systems naturally drift toward disorder unless energy is actively used to maintain their structure, and that means your existence is not passive. It is an ongoing process of holding together something that would otherwise come apart. Inside your body, at every second, trillions of microscopic interactions are taking place, enzymes scanning and repairing damaged DNA before errors can accumulate, proteins folding into precise configurations required for function and being corrected when they misfold, cellular structures being rebuilt and replaced as they degrade, and gradients of ions being maintained across membranes so that cells can continue to generate signals and sustain life. And none of this happens automatically in the sense of being free, because every one of these corrections, every act of maintenance, every small victory against disorder requires energy, specifically ATP, the molecular fuel that powers nearly all biological work, and your body produces and consumes astonishing amounts of it just to preserve the pattern that defines you. But here is where the system reveals its limitation, because that energy is not infinite and cannot be used everywhere at once, which means your body must constantly decide how to allocate it, whether to invest it in maintenance and repair, preserving the integrity of tissues and molecular structures over time, or to divert it toward immediate survival, preparing for threats, mobilizing resources, increasing vigilance, and shifting into a state where short-term performance takes priority over long-term stability. And while those two modes can coexist briefly, they cannot both be maximized simultaneously, because they draw from the same finite pool. Under normal conditions, this balance is dynamic and adaptive, with the system shifting into defense when needed and then returning to repair once the threat has passed, allowing accumulated damage to be corrected and stability to be restored. But the problem emerges when the signal that drives this shift does not turn off, when the body interprets the environment as persistently threatening, even if that threat is not physical or immediate. Because in that case, the allocation changes in a way that is subtle but relentless, with more energy being continuously diverted toward defense and less being available for maintenance, not enough to cause immediate failure, but enough to gradually reduce the efficiency of every system responsible for preserving order. What follows is not a dramatic breakdown, but a slow drift, a gradual accumulation of small imperfections that are no longer fully corrected, proteins that remain slightly damaged, DNA lesions that are repaired less efficiently, cellular components that are replaced more slowly, and structures that begin to lose their precision. And because biological systems depend on continuous correction to maintain low entropy states, even a small reduction in repair capacity leads to a compounding effect over time, where minor deviations reinforce one another and the overall integrity of the system declines. At that point, aging stops being an abstract concept tied to the passage of years and becomes something much more concrete, a measurable outcome of how effectively your body continues to invest energy into maintaining its structure, because the rate at which disorder accumulates is not fixed, but depends directly on how much energy is available for correction. And when that investment is reduced, even slightly but persistently, the balance shifts in favor of entropy. not as a sudden event, but as a continuous process that unfolds quietly beneath your awareness. And if you follow that logic all the way through, you arrive at a realization that is difficult to ignore, because it means that the difference between maintaining the structure that is you and gradually losing it is not determined only by time or genetics, but by how your body allocates its energy in response to the signals it receives, signals that can keep it in a state of readiness long after any real danger has passed, slowly redirecting resources away from the very processes that keep you intact, until the system is no longer fully sustaining itself at the level it once did. And from that moment forward, the trajectory is no longer neutral, but already leaning, almost imperceptibly, toward disintegration. Now, if what we just talked about is true, if your body is constantly deciding how to spend a limited supply of energy between maintaining itself and preparing for danger, then the next question is unavoidable, because something has to be making that decision. Something has to be reading the situation and telling every cell in your body whether this is a moment to repair or a moment to survive. And that role belongs to a single chemical signal that moves through your bloodstream and quietly rewrites the priorities of your entire system. A molecule your own body produces, not occasionally, not in rare circumstances, but every single day. And that molecule is cortisol. Cortisol is not a villain. And this is important to understand from the beginning, because in the short term, it is one of the most useful substances you have. It is synthesized from cholesterol in your adrenal glands and released when your brain detects a threat, real or imagined, through a cascade that begins in the hypothalamus, passes through the pituitary gland, and ends in the adrenal cortex. And when it enters your bloodstream, it triggers a set of changes that are remarkably efficient. Your liver begins converting amino acids and glycerol into glucose so that your muscles and brain have immediate fuel. Your attention sharpens as certain regions of your brain become more sensitive to incoming information. Inflammation is temporarily suppressed so that minor injuries do not interfere with performance. Blood flow is redirected away from digestion and toward the organs that matter most in an emergency. And processes that are not immediately necessary, such as reproduction and long-term tissue building, are placed on hold. Everything about this response is optimized for urgency, not duration. And under normal conditions, it operates as a precisely regulated system. Cortisol rises quickly in response to a challenge and then falls just as quickly once that challenge is resolved, guided by a feedback loop in which cortisol itself signals the brain to stop producing more of it. A classic example of negative feedback similar to a thermostat that turns off heating once the desired temperature is reached. And as long as that loop is functioning properly, the system remains stable, shifting into action when needed and returning to baseline afterward. But the entire structure depends on sensitivity, because the brain must be able to detect when enough cortisol is present to shut the system down. And this is where chronic stress changes the rules, because when cortisol levels remain elevated for extended periods, the receptors that detect it begin to downregulate, becoming less responsive to the signal they are supposed to read. And as a result, the feedback loop weakens. The brain no longer receives a clear message that cortisol is already high. And so it continues to drive production even when it should stop, creating a system that is no longer self-correcting but drifting, overshooting its set point and failing to return. In control theory, this is what happens when a feedback system loses gain in its regulatory pathway. The restoring force disappears, and the system stabilizes at a higher level than intended, not because it is designed to operate there, but because it no longer has the sensitivity required to bring itself back. And in the case of cortisol, that new level is not extreme enough to trigger immediate alarm, but it is persistent enough to alter the chemistry of your body every single day, flattening the natural rhythm that should rise in the morning and fall at night, keeping levels slightly elevated when they should be low and insufficiently dynamic when they should fluctuate. This creates a state that is easy to overlook because it does not feel like an emergency. There is no dramatic surge, no clear signal that something is wrong, but beneath that surface, every cell in your body is receiving a consistent instruction: prioritize survival, redirect energy, hold resources in reserve. And over time, that instruction becomes the default setting rather than the exception, shifting the entire system away from repair and toward defense, not as a temporary adjustment, but as a chronic condition. And this is the point where the logic from before becomes unavoidable. Because if maintenance requires energy, and that energy is being continuously diverted, then the processes that preserve your structure begin to slow down. Not abruptly, not catastrophically, but just enough to allow small forms of damage to accumulate, just enough to let entropy gain a slightly stronger foothold. And once that shift happens, the system is no longer balancing itself in the way it was designed to, because the real problem is not that cortisol rises when you need it. The problem is that the system forgets how to turn it off. Once that system shifts, once cortisol remains elevated longer than it was ever designed to, the consequences do not stay confined to hormones or abstract regulation. They begin to translate into physical chemistry, into reactions that unfold slowly but relentlessly inside your tissues. And one of the most important of those reactions is something so ordinary that you have seen it countless times without realizing that it is happening inside your own body, because it is the same process that browns bread in a toaster, that gives a seared steak its crust, that turns onions golden when heat is applied over time. And in your body, that same chemistry is running quietly at 37 degrees Celsius. Not in minutes, but over years, gradually altering the very structure that holds you together. Cortisol's primary metabolic role is to increase the availability of glucose in your bloodstream, ensuring that energy is immediately accessible when needed. And while this is useful in short bursts, the problem begins when glucose remains elevated for extended periods, because glucose is not an inert molecule simply floating harmlessly through your blood. It is chemically reactive, and when it encounters proteins, especially long-lived structural proteins like collagen, it can bind to them in a process known as glycation, forming new molecular structures that were never part of the original design. Collagen is not a minor component of your body. It is the most abundant protein you have, making up roughly a third of your total protein content, forming the scaffolding of your skin, the tensile strength of your tendons, the flexibility of your cartilage, the integrity of your blood vessels, and even contributing to the clarity of your eyes. And what makes collagen particularly vulnerable is not just its abundance, but its longevity. Because unlike many proteins that are constantly broken down and replaced, some collagen structures persist for years or even decades, meaning they are exposed to circulating glucose for extraordinarily long periods of time. The chemistry begins simply enough with a glucose molecule colliding with an amino group on the surface of a protein, forming an unstable bond that rearranges into a more stable intermediate. And over time, through a series of further reactions, this process produces what are known as advanced glycation end products, or AGEs, molecules that can attach to proteins in ways that fundamentally alter their properties. And the most damaging of these do something particularly problematic. They create permanent cross-links between adjacent protein strands effectively binding them together in a way that the body cannot undo. This is where the process becomes irreversible, because while your body has evolved enzymes capable of repairing many forms of molecular damage, there is no biological mechanism capable of breaking these glycation-induced crosslinks once they form. The energy required to reverse them exceeds what your cellular machinery can generate under normal conditions, which means that each crosslink represents a permanent change in the structure of your tissue, a small but cumulative shift toward stiffness, toward brittleness, toward reduced function. Over time, as these crosslinks accumulate, the effects become visible and measurable. Collagen in the skin loses its elasticity, making it less able to stretch and return to its original shape, leading to the formation of wrinkles. Blood vessels become less compliant, reducing their ability to absorb the pressure of each heartbeat and contributing to increased blood pressure. Joints lose their smoothness and flexibility as cartilage stiffens and degrades. And even the lens of the eye can become clouded as structural proteins lose their transparency. What makes this process particularly striking is not just that it happens, but that it happens slowly enough to go unnoticed while it is occurring, because no single glycation event changes how you feel. No single crosslink produces pain or dysfunction. But as they accumulate over years, the cumulative effect becomes undeniable. A gradual transformation of the physical properties of your tissues, driven not by external damage, but by internal chemistry, by a reaction that is continuously fed by the presence of glucose. And when you trace that back to its origin, to the elevated glucose levels maintained by chronic cortisol signaling, you begin to see how a regulatory shift at the hormonal level translates into structural changes at the molecular level. How a decision about energy allocation becomes a chemical modification of the proteins that define your physical form, and how that modification, once made, cannot simply be reversed by returning to baseline. Because this is not wear and tear in the usual sense, it is your body very slowly being altered in a way that it cannot fully undo. If glycation shows you how stress reshapes the physical structure of your tissues over time, then telomeres reveal something even more unsettling, because they show you how stress reaches deeper than proteins and begins to mark time directly into your DNA. Not metaphorically, not as an abstract idea of aging, but as a measurable, quantifiable shortening written into the very ends of your chromosomes every time a cell divides. A process so precise that it can be tracked, compared, and accelerated under the right conditions. At the end of every chromosome in your body, there are repeating sequences of DNA. Simple patterns that do not code for any protein, but exist purely as protective buffers, absorbing the loss that occurs during cell division, because the machinery that copies DNA cannot fully replicate the very ends of each strand. And as a result, with every division, a small portion of that telomeric sequence is lost, a few dozen base pairs at a time, ensuring that the essential genetic information remains intact while the expendable ends gradually shorten. Under normal conditions, this shortening is part of a controlled system, a kind of biological clock that limits how many times a cell can divide, known as the Hayflick limit, preventing damaged or aging cells from replicating indefinitely. But this process is not fixed in its rate, because the length of telomeres does not decrease at a constant speed across all individuals or even across all circumstances within the same individual. And that is where stress enters the picture in a way that fundamentally changes the trajectory. Chronic exposure to stress hormones particularly cortisol, combined with increased oxidative stress within the cell, accelerates the rate at which telomeres shorten, not by altering the basic mechanism, but by increasing the damage that accumulates during each cycle of replication. And at the same time, it suppresses the activity of telomerase, the enzyme responsible for rebuilding and extending these protective sequences, meaning that not only are telomeres being lost more quickly, they are also being restored less effectively. Shifting the balance toward depletion, what makes this more than just a biochemical detail is that telomere length is directly linked to cellular function, because once telomeres become too short, cells enter a state known as senescence, where they stop dividing and begin to secrete inflammatory signals that affect surrounding tissue, contributing to systemic aging. And when enough cells reach this state, the cumulative effect becomes visible at the level of the organism, not as a single failure, but as a gradual decline in the ability to repair, regenerate, and maintain stability. This is not theoretical. It has been measured in human populations under conditions of chronic stress, where individuals exposed to prolonged psychological strain show telomere lengths equivalent to those of people significantly older, sometimes by as much as a decade, meaning that their cells have effectively aged faster than their chronological time would suggest, compressing years of biological wear into shorter periods simply through the sustained activation of stress-related pathways. But here's where the story takes a turn that is easy to overlook if you are only following the mechanism and not the implication. Because the acceleration of telomere shortening is not purely determined by external conditions, it is mediated by how those conditions are interpreted, by the signals the brain generates in response to them. And this has been demonstrated in cases where individuals exposed to similar levels of objective stress show markedly different biological outcomes, with some maintaining relatively stable telomere lengths, while others exhibit significant shortening. That difference cannot be explained by the environment alone, because the external situation is the same. And it cannot be fully reduced to genetics, because the variation appears within comparable populations, which means that the variable influencing the rate of cellular aging is not only what happens to you, but how your system processes what happens, how it translates experience into biochemical signals that either amplify or dampen the mechanisms that drive telomere loss. And when you follow that logic all the way down, you arrive at a conclusion that challenges the way most people think about aging. Because it suggests that time, at least at the cellular level, is not simply something that passes uniformly, but something that is written at different speeds depending on the internal state of the system, a state shaped by stress, perception, and the signals that determine whether the body remains in a mode of sustained alertness or is allowed to return to repair. So you can have two people living through the same reality, exposed to the same demands, the same pressures, the same duration of stress, and yet at the level of their chromosomes, at the level where time is physically recorded, one of them is aging faster than the other, not because more time has passed, but because more of it has been written into their cells. If telomeres show you that stress can change how fast time is written into your cells, then the next layer goes even further, because it reveals that the signal driving all of this does not stop at damage or depletion. It reaches into the system that decides which parts of your DNA are even used in the first place, effectively reprogramming the behavior of your cells without altering the underlying genetic code. And the trigger for that shift is not physical injury, not infection, but something far less tangible and far more pervasive: the perception of social isolation. Your DNA, all 3 billion base pairs of it, remains essentially the same in every cell of your body. But what matters for function is not just the sequence itself. It is which genes are turned on and which are turned off at any given moment, a process known as gene expression, controlled by networks of regulatory proteins that respond to signals from the environment, translating those signals into patterns of activity that determine what the cell produces, how it behaves, and how it contributes to the larger system under conditions where the brain interprets the environment as safe, supported, and socially connected. Gene expression follows a pattern that prioritizes balance, repair, and efficient immune function. But when that perception shifts, when the system begins to interpret the social world as threatening, unpredictable, or isolating, a different program begins to run, one that has been conserved across evolution because of its survival value in a very different context from the one you live in now. This program, identified in multiple studies, is known as the conserved transcriptional response to adversity, and its signature is both specific and consistent. Genes associated with inflammation are upregulated, increasing the production of molecules that drive swelling, tissue response, and aggressive immune activity, while at the same time, genes associated with antiviral defense are downregulated, reducing the body's ability to respond to viral threats. And this shift is not random. It reflects an ancient survival strategy in which isolation from a group increased the likelihood of physical injury and bacterial infection while reducing the likelihood of viral exposure from close social contact. In that context, prioritizing inflammation over antiviral defense made sense. It increased the chances of surviving wounds, of preventing bacterial invasion, of responding quickly to immediate physical danger. But in the modern environment, where the threats associated with isolation are no longer primarily physical, this same program becomes maladaptive, creating a state of chronic low-grade inflammation that persists without a clear target, gradually damaging tissues and contributing to a wide range of conditions, including cardiovascular disease, metabolic disorders, depression, and neurodegeneration. What makes this mechanism particularly striking is that it is not driven by objective measures of social contact. It does not depend on how many people you interact with, how often you are around others, or even whether you live alone or with a group. It depends on perception, on whether your nervous system registers those interactions as safe, supportive, and meaningful, because the signal that drives gene expression originates not from external reality itself, but from the brain's interpretation of that reality. This means that two individuals can exist in nearly identical social environments and yet display completely different gene expression profiles, one showing the inflammatory signature associated with isolation, while the other does not, simply because one perceives connection where the other perceives threat. And that difference is not abstract. It is measurable at the level of immune cell production, where stress-related signaling pathways stimulate the release of immature white blood cells that are predisposed toward inflammatory responses, altering the composition of the immune system itself. At the molecular level, this shift is coordinated by specific transcription factors, regulatory proteins that bind to DNA and determine which genes are expressed, with pro-inflammatory pathways becoming more active and antiviral pathways becoming less so, effectively redirecting the immune system toward a mode that is prepared for a type of danger that may never actually occur, while simultaneously weakening defenses against the types of threats that are more relevant in the present environment. And when you step back and look at the system as a whole, what becomes clear is that this is not simply a psychological effect translated loosely into biology. It is a precise form of signal processing, where the brain acts as an interpreter, converting social experience into biochemical instructions that reshape cellular behavior, meaning that the state of your immune system is not determined solely by pathogens or physical damage, but by the signals your brain sends about whether you are safe. So what you have is a system that does not respond to how many people are around you, but to whether you feel protected by them. A system that can place your body into a state of sustained inflammatory readiness, not because of any external threat, but because of an internal assessment that something is missing, something that evolution has treated for millions of years as a condition that must be corrected immediately. And that leads to a transition that is easy to miss if you focus only on the biology, because once the signal of threat is established at the level of perception and translated into gene expression, it does not remain isolated within the immune system. It propagates outward, influencing the nervous system, the endocrine system, and the allocation of energy itself, setting the stage for a deeper shift in how the entire body regulates the balance between defense and repair. Once you understand that perception can reprogram gene expression, that the brain can shift the immune system, the endocrine system, and even the rate at which your cells age, simply by interpreting the environment as safe or threatening, then the next question becomes unavoidable. Because if a signal can push the body into a prolonged state of defense, there must also be a signal capable of bringing it back, a mechanism that tells the system that the danger has passed, that resources can be reallocated, that repair can resume, and that signal does exist, but it is not abstract. It is not philosophical. It is biochemical, measurable, and deeply tied to something as simple and as specific as human connection. At the center of this shift is a small peptide hormone called oxytocin, produced in the hypothalamus and released into the bloodstream and brain in response to certain types of social interaction, particularly those involving trust, physical touch, eye contact, and emotional warmth. And what makes oxytocin remarkable is not just that it is associated with bonding or attachment, but that it directly counteracts many of the physiological processes driven by chronic stress, lowering cortisol levels, reducing inflammatory signaling, and enhancing the efficiency of immune responses in ways that can be observed and measured. But oxytocin does not act alone. It is part of a larger system that governs how your body transitions between states of alertness and recovery. And that system is centered around the vagus nerve, the primary pathway of the parasympathetic nervous system, which extends from the brainstem through the heart, lungs, and digestive organs, acting as a kind of communication channel that regulates heart rate, digestion, immune function, and overall physiological calm. And when this system is active, the body shifts away from defense and toward maintenance, slowing the heart, lowering blood pressure, reducing cortisol production, and reallocating energy toward processes that repair damage and preserve structure. The activity of the vagus nerve can be observed indirectly through heart rate variability, the subtle variation in the time interval between heartbeats, which reflects the system's ability to adapt flexibly between states, and higher variability is associated with greater resilience, better health outcomes, and a stronger capacity to recover from stress, while lower variability is linked to chronic stress, inflammation, and increased risk of disease, making it one of the most informative indicators of how well the body is balancing the competing demands of survival and repair. What ties all of this together is the fact that social connection directly influences this system, not through abstract feelings alone, but through specific sensory inputs, the sound of a calm and familiar voice, the expression on another person's face, the physical sensation of touch, all of which are processed by neural circuits that are linked to the vagus nerve, allowing the brain to rapidly assess whether the environment is safe. And when that assessment is positive, the vagus nerve increases its activity, shifting the entire body into a state where repair processes are prioritized. This is not a gradual or symbolic effect. It can occur quickly and produce measurable changes, with cortisol levels decreasing, inflammatory markers dropping, and immune function improving in response to supportive social interaction. And studies have shown that even small differences in the quality of those interactions can significantly alter physical outcomes, such as the rate at which wounds heal, demonstrating that the body's capacity to repair itself is directly influenced by the signals it receives about safety and connection. What is particularly important here is that the effect depends on authenticity, because the nervous system is not responding to the idea of social contact, but to the actual experience of safety. Meaning that superficial interaction or forced connection does not produce the same biological response, while genuine trust and warmth do, triggering the release of oxytocin and activating the vagus nerve in a way that allows the system to stand down from its defensive posture. When that shift happens, the consequences extend across multiple levels of the body. Cortisol decreases, which reduces blood glucose and slows damaging chemical reactions like glycation. Telomerase activity can increase, supporting the maintenance of chromosome integrity. Inflammatory gene expression is reduced, restoring balance to the immune system, and energy that was previously held in reserve for defense becomes available again for repair, allowing the molecular systems that maintain your structure to function more effectively. And if you trace all of those changes back to their origin, what you find is not a complex intervention or a technological solution, but a change in the signal that your body is receiving about the world, a shift from threat to safety that reactivates systems that were never broken but simply deprioritized, waiting for the right conditions to resume their work. So what brings your body back is not the absence of stress in some absolute sense, but the presence of something that tells your system it is no longer alone in dealing with it. If all of this seems disproportionate, if it feels almost unreasonable that something as intangible as social connection could influence hormones, gene expression, immune function, and even the rate at which your body accumulates damage, then the only way to make sense of it is to step back and look at the conditions under which this system was built. Because none of these mechanisms evolved in the world you are living in now. They were shaped over hundreds of thousands of years in an environment where being alone was not a psychological inconvenience, but a direct and immediate threat to survival. For the vast majority of human history, individuals did not exist as isolated units, navigating independent lives. They existed as members of tightly bonded groups where cooperation was not optional but essential, where survival depended on shared vigilance, shared resources, and shared protection. And in that context, separation from the group dramatically increased the probability of injury, starvation, or death, not eventually, but quickly, which meant that any biological system capable of detecting and responding to isolation had a direct evolutionary advantage. What emerged from that pressure was a nervous system that treats social disconnection as a critical signal, one that activates the same pathways involved in physical danger, increasing vigilance, elevating cortisol, shifting the immune system toward inflammatory readiness, and reallocating energy toward immediate survival, all in an effort to prepare the individual for the risks associated with being alone in a hostile environment. And in the world in which these mechanisms evolved, that response was not only appropriate, it was necessary, but the environment has changed far more rapidly than the biology that responds to it. Because the modern world allows for physical safety without social connection, making it possible to be alone without immediate external threat, while the internal systems that interpret isolation have not been updated to reflect that change. And as a result, the body continues to run an ancient survival program in a context where its assumptions are no longer valid, maintaining a state of readiness that is no longer adaptive but becomes damaging over time. This mismatch between environment and biology creates a situation where the absence of connection is processed not as a neutral condition, but as a persistent signal of danger, keeping stress pathways active, maintaining elevated cortisol, sustaining inflammatory gene expression, and continuously diverting energy away from the processes that preserve long-term stability. And because these changes occur gradually and without a single defining moment, they are often experienced not as a direct consequence of isolation, but as a diffuse decline in physical and mental resilience. The scale of this effect becomes clear when you look at population-level data, where large studies have shown that individuals with strong social relationships have significantly higher survival rates over time compared to those who are socially isolated, with differences that are comparable to well-established risk factors such as smoking or lack of physical activity, indicating that social connection is not simply beneficial, but fundamentally tied to the probability of survival itself. What makes this particularly significant is that the relationship follows a consistent pattern where increasing levels of isolation correspond to increasing levels of risk, suggesting that the effect is not incidental, but reflects an underlying mechanism that operates across different contexts and populations, reinforcing the idea that the body is responding to social disconnection in a way that has measurable consequences for health and longevity. And when you bring all of these layers together, from energy allocation and hormonal regulation to molecular damage, gene expression, and evolutionary history, a coherent picture begins to emerge, one in which social connection is not an optional aspect of well-being, but a variable that directly influences the physical processes that determine how long and how well the body can maintain its structure against the constant pressure of entropy. So the question is no longer whether connection matters in some abstract or emotional sense, but whether the signals your body is receiving are allowing it to remain in a state where repair is possible, where resources are allocated toward preservation rather than defense, where the systems designed to maintain you are given the conditions they require to function as intended. Because if the absence of connection can shift your biology in measurable ways, if it can alter how your genes are expressed, how your immune system behaves, how your energy is distributed, and how quickly your cells accumulate damage, then what does it mean, in strictly physical terms, to continue living in a state where those signals never fully change?